Emily, and I'm a PhD candidate in the DECUS lab at Stanford University, and I study the limits of life in hypersaline environments. So as we all probably know, brines are one of the targets for a life detection mission to other ocean worlds, including Europa and Enceladus, and even past ocean worlds like Mars. And so by understanding the limits of salty life on Earth, we can understand how and where to look for life on these other places. One of the ways we can assess a brine environment is through this measurement called water activity. And water activity depends on vapor pressure, but you can think of it as how many water molecules are available to a cell in solution. So on the left is an image of what pure water would be. This is where all the water molecules are free to become vapor, move about the cabin. That would be a water activity of 1. Now, if there's ions in solution, water would be attracted to those molecules and a water activity would decrease. So the lower the water activity, the more uninhabitable an environment is. And since water is required for all life as we know it, water activity can be used to assess habitability. So some common items and their water activities are listed on the left, including seawater, which has a pretty high water activity, and the salinity of seawater is about 3.5%. And then honey has a water activity of 0.6, and some of Mars' special regions or areas where we might detect life on Mars are no lower than 0.5. And there's been a lot of really great pure culture work on the limits of life and cell division at different water activities. And to sum it up, the predicted or theoretical water activity limit for all life on Earth, including eukaryotes and prokaryotes, is about 0.6. And this is based on pure culture, so like our speaker last night mentioned, not all the things in the world are in pure culture yet, so that leaves out about 90% or more of the biodiversity on Earth, and our ability to assess what the limit of life for each of those strains of microbes are. So brine environments are really widespread on Earth. They're everywhere. And a lot of great work has gone into assessing metagenomics, like the taxonomic diversity, the potential, metabolic potential of microbes in these environments, metatranscriptomes, and even some bulk activity measurements. But metabolic activity is rarely measured at the single cell level. And this can be really important because when a cell is stressed out, they might have really low levels of activity that are not detected with bulk techniques. So today I'll be talking a little bit about my work in solar salterns and then transition into my current work in Western Australia acidic brines. So, like I mentioned, when life is reaching its limit, metabolic activity can be really low. And so single cell analysis can help us detect this low level of activity by lowering detection limits. So a cell needs to acquire 50% new biomass to be able to divide into 2 new daughter cells. However, with Nano SIMs, for example, we only need to detect about 5% maximum. We can even get as low as less than 1% of new biomass to detect active life in a sample. It also provides quantitative results, so we can do a lot with this data on a quantitative level, and it also reveals trends that are often obscured by bulk analysis. And when I'm talking about metabolic activity in this talk, I'm talking specifically about anabolic activity. So there's catabolic activity that produces energy for a cell, and then there's anabolic activity where a cell can take substrates from the environment, like amino acids and glucose, and take that carbon and nitrogen, and then produce new cell biomass, which includes proteins, DNA, and cell walls, and that sort of thing. So first, the work that I've done at this solar saltern, it's called South Bay Saltworks. It's here in San Diego. And basically, they bring seawater in from San Diego Bay, and it goes through a series of evapoconcentration ponds. And as the water evaporates out of the seawater, different salts are left behind, producing a series of ponds that range in water activity from 0.98, that of seawater, to that well below the known limit of life at 0.4. And so we measured anabolic activity of over 6,000 individual cells from a series of 5 of these brines with 5 different isotopic substrates from amino acids, ammonium and bicarbonate, glucose and nitrate, and that sort of thing. We measured their anabolic activity with Nano SIM. So on the left, this is what this looks like. Cells that are circled in red are considered enriched, and this is for carbon from amino acids. And then the cells that are circled in white would be considered less active or less enriched in this sample. And when you plot this with water activity, you see that microbial activity decreases exponentially with water activity. However, there's certain areas, let's see, can we see this? Yeah. certain subsets of cells are more active than most of the cells in seawater at a higher water activity, with even just low decreases in water activity. We also detected cell biomass through DAPI and Nano SIMs in this low water activity magnesium chloride brine. However, we didn't detect any activity here. And the incubation time was 2 days long, so there's several reasons for this. We were also able to use this data to estimate a new predicted lower limit of life at 0.54, so that's a little significantly, biologically significantly lower than the 0.6 previous limit predicted. And this data, I'd love to talk more about it, but it's been submitted, and I just got word that it's now in review as of this morning, so that's really exciting. And if you'd like to hear more about that, please talk to me later. So now to kind of get into the topic of this talk, which is talking about the limits of life in acidic brine environments. There's a lot of things that happen in brines. So you can have salinity as a limiting factor, you can have high metal concentrations, you can have high ionic strength and chaotropicity. But one thing that really stood out to me about this sample set was that most of the brines that had high levels of activity actually had a pH about 7 or 8, so like relatively neutral to slightly basic pH. However, this magnesium chloride brine, which had no detectable activity, had a pH of about 5.4. And so my question was, how does pH affect microbial activity in brine environments? And is this an important factor in the limits of life? So last summer, our team went to Western Australia to sample a bunch of acidic brines. And these brines are considered Mars analogs because of the geochemistry and weathering systems that created them over millions of years. Just in short, oxidation of sulfides acidified the water, and then evaporation concentrated ions and other salts over time, creating these really acidic briny environments. There's hundreds of these lakes, and we were really lucky to sample a really broad spectrum of them. So on the x-axis is water activity, and on the y-axis is pH. And I was really thrilled to get a nice group of lakes to sample for activity analysis, especially these ones that are boxed. These were at a water activity of 0.7, and they range in pH. So it's a really good opportunity to kind of look at how pH affects metabolism at low water activity. So with this study, I analyzed metabolic activity, specifically anabolic activity, by incubating these brines with an analog amino acid called HPG. Cells that are active in the sample will take up HPG and incorporate them into their new proteins that are produced. And then the HPG has a alkyne group on the end that later on in lab you can perform click chemistry on and click a fluorophore to it. So all the cells that are active in the sample turn out green under the microscope or in flow cytometry, and then that can be counterstained or compared to cells that are stained blue via DNA stain of DAPI. And so with this data, you can calculate the percent of active cells across these lakes. So this is just an example of what this data set looks like. These are just 4 of the 13 or 14 lakes that we were able to do this with. On the left, the lakes are ordered in decreasing pH. And I thought it was also kind of interesting, the color changes, so you can admire those. I don't know why they do that, but that's the way it is. And then in the middle are the DAPI stained cells under the microscope, and then on the right are the active cells that are tagged with the green fluorophore. And you can also notice like the different cell morphologies and abundances that change with pH, which is really fascinating. And also that pH didn't affect the assay itself. We do see some cells that are active at these low pHs, and sometimes you have brines where there's less activity happening there. So when you calculate the percent of active cells and you put this all in a plot, I'll just orient you. So on the x-axis here, we have water activity decreasing, and on the y-axis is pH decreasing in this direction. So you would expect the most extreme acidic brines to be kind of in this region. This dotted line is that newly predicted water activity limit of life that I talked about earlier. And then this dotted line represents the percent of active cells dropping below 50% at this pH. So this color bar, each of the dots is one lake, and the color bar was colored on a green gradient if the percent of active cells is above 50%, and on a gradient towards black if it was below 50%. So you can kind of just visually see that all of the green, like high percentage active cells cluster up here, and all of the low percent of active cells cluster down here. And when you look at it statistically, there is a moderately stronger correlation with pH and activity compared to water activity and activity. So many times, you can count how many times I say activity in this talk. And so some thoughts on this. A pH of 6 is really high for environmental samples. And as we've heard from a lot of people, and as we know, there's a lot of microbes that live in way more acidic environments. So what's actually happening here, I don't have an answer to that yet. And I'm hoping to figure that out with future analyses that include Nano SIM's work on these lakes. And then if this is a true signal and this is what is going on in the environment, then microbial activity may be detected at lower water activities than we know of now, as long as the pH is within the 7 to 8, maybe like not so acidic range. So figuring out what that pH range is and how that interacts with water activity limits of life is something of ongoing investigation. So with that, I'd like to acknowledge my lab, the DECAS lab pictured up in the right, all of our funding sources, the Oceans Across Space and Time team, which is a NASA-funded project you'll hear about. You'll hear from a lot of us throughout this conference, and all of our collaborators in Western Australia, including Molly the dingo, who, of course, this work would not have been possible without. All right, and then I'll take any questions, and I'll leave this up so you can stare at the dots for a little bit longer.
Thank you, Emily. Any questions?
Hi, that was a really cool talk. Thanks so much. I was just wondering if you could expand a little bit on why you concentrate only on anabolic activity as opposed to catabolic activity.
Yeah, so let's see. Scientifically, the reason why I would focus on anabolic activity, I mean, both catabolic and anabolic activity are essential for life, right? Life has to make energy to assimilate substrates and grow and divide. So both processes are really important. And they are processes that I'll be combining or looking at in future field sites. With anabolic activity, the interesting thing here is that you could imagine that when a cell gets really stressed out, it would stop dividing potentially and still accumulate substrates to repair DNA, repair cell walls, do all that stuff until conditions get better. And so the idea was that, or my original hypothesis was that that anabolic activity would extend beyond the known cell division limit of life, because even as cells would be able to survive just by taking up materials. And so that taking up process is the anabolic activity part. Yeah, good question.
Sweet talk. I have a question. So you mentioned that you guys have essentially extrapolated a new limit for life, in terms of water activity. It's kind of a power move. So
we're not the first ones to do this.
Okay, but could you explain how you extrapolated that? Because I think maybe I missed that or I didn't understand.
Yeah. Okay, we're going to get into the weeds here, but let me go back to that slide. So this idea or this extrapolation is based on this study here. So they did the same thing, and this is how they got this theoretical limit of life that has now been cited almost 200 times from food preservation fields to life detection fields. So
yours is lower.
hasn't been published yet. So they used a different technique, and they based it on cell division. And so so hang on, let me answer your first question. So the way that we did this is single cell analysis allows you to separate the cells that are active from those that are inactive. And in a brine environment, this is really important because brines are really salty and cells can fall in from anywhere, bird poop, wind, whatever, and get preserved, and those can be inactive. So those can contribute to that bulk analysis and not necessarily even be important in that environment. So with single cell analysis, we took out the active cells, averaged their anabolic activity, so we're just focusing on who's contributing to metabolism in that environment, and then use detection limits and math to extrapolate and predict or propose this new predicted limit of detectable life. And so this is not the limit of life. This is what our measurements can detect. And if we found an environment at 0.54 water activity with similar conditions to this environment that we extrapolated it from, we would technically be able to detect life in that environment. And this is also a really conservative limit. So we even upped the number of standard deviations that we would normally do for our detection limit. So if we were to do our data processing in the normal way, it might even be lower. That kind of helps. Yeah. Yeah. Mm-hmm.
Hi, George Scheibel from Montana State. I'm just curious, you're looking at the active cells. Did you look at who they were at all, whether that was by fish or sequencing?
So in this study, we did not look at cell identity, and we focused on metabolism. In Western Australia, I don't have plans to look at the, I don't know, I could look at the active fraction of cells from the Boncat analysis. I haven't decided if I want to go down that road yet, but in the next field site, we are planning on doing that. So yeah, not in this study.
All right, thank you very much, Emily.
Yeah, thank you.